Alright, back to this overview of fat metabolism. We have now covered the catabolic pathways, namely the degradation of triacylglycerols to fatty acids and the degradation of fatty acids to acetyl-CoA via beta oxidation. We also discussed the production of ketone bodies and their importance when glucose is unavailable. Now, I want to consider the anabolic side of fats. The two main pathways for this are the synthesis of fatty acids from acetyl-CoA and the production of triglycerides from fatty acids. Throughout this section, I will also discuss a bunch of regulatory mechanisms that are in place to control the degradation and synthesis of fats. So, to get started, we'll begin with fatty acid synthesis, but before we jump into the reactions, I want to first consider some intuition on fat anabolism. As you can see in this overview of fat and carbohydrate metabolism, acetyl-CoA is a converging metabolite for both glucose and triglycerides. This is super important because it ties the two nutrients together, and a crucial implication of this is that excess glucose can be stored under the form of fat. Indeed, glycogen, which stores glucose, is only utilized by a select amount of tissues, but fat storages can be dispersed across the body in a relatively unlimited quantity. Now, since acetyl-CoA is at the crossroads between many anabolic and catabolic pathways, it's important that for it to be used in fat synthesis, the body is in a state of excess energy to promote anabolism. As we've seen in the regulatory mechanisms of other pathways, whether it is glycolysis or the citric acid cycle, the needs of the cell always dictate what it does with its nutrients. So let's assume that the glycogen stores are filled up and we still have a massive quantity of glucose and other sugars to process. These carbohydrates will be consumed by glycolysis and the pyruvate dehydrogenase complex and eventually produce acetyl-CoA. Normally, the acetyl-CoA would be processed by the citric acid cycle, but if we take a look at the cycle in more detail, when NADH and ATP accumulate, it shuts down all the rate-limiting steps. Recall that NADH is one of the main products of the cycle and is used by the electron transport chain to make energy. Since we don't need energy, the electron transport chain shuts down and thus causes the accumulation of NADH, which shuts down all the catabolic pathways that feed into the citric acid cycle. So, to avoid having the acetyl-CoA accumulate and become toxic to the cell stores it in the form of fatty acids. Hopefully, with this you can see how glucose is related to fat synthesis. With all this being said, an important aspect about fat synthesis is that it happens in the cytosol, but the acetyl-CoA produced by the degradation of carbohydrates or fats occurs in the mitochondrion. As such, the first step to consider in this entire process is how acetyl-CoA is shuttled out of the mitochondrion. In comparison to some of the other shuttles we covered in previous discussions, the transport mechanism here is pretty straightforward. Basically, the acetyl-CoA that is made in the mitochondrion first gets integrated in oxaloacetate to produce citrate. This reaction is also the first step of the citric acid cycle. Since the cycle is now stopped due to energy excess, the mitochondrion has transporters that allow citrate to leave and join the cytosol. In there, citrate can be broken back to acetyl-CoA and oxaloacetate in a reaction catalyzed by ATP citrate lyase. This reaction requires the hydrolysis of ATP to happen. The oxaloacetate that is made from this reaction can be returned to the mitochondrion via malate, as the mitochondrion has transporters for it. To convert between the two, the enzyme malate dehydrogenase uses NADH in the cytosol to make malate and NAD+ to regenerate oxaloacetate in the mitochondrion. As a quick aside, if you recall the regulation of glycolysis, you might remember that citrate was an inhibitor to phosphofructokinase 1. The logic for this regulation was that basically, since we are in a state of high energy, as indicated by the presence of citrate in the cytosol, there is no need for glycolysis to keep going and degrading glucose. With this being said, now that we have acetyl-CoA in the cytosol, we can begin covering how fatty acids are made out of it. So, for this entire process, there are basically two enzymes that mediate the synthesis: acetyl-CoA carboxylase and fatty acid synthase. Acetyl-CoA carboxylase is responsible for one reaction, which is the addition of a CO2 group on acetyl-CoA to produce malonyl-CoA. This reaction requires the hydrolysis of ATP to happen, and as a little foreshadowing note for later, this is the rate-limiting step that receives all the regulation. The second enzyme, fatty acid synthase, is a multi-complex protein that performs the remaining reactions to get fatty acid. To get an idea of the protein, here is a surface representation that shows the multiple subunits. From the symmetry of the protein, you can notice that fatty acid synthase is a dimer. The implication of this is that the protein is able to produce two fatty acids at the same time, but this won't be too much of a concern for us. Now, in comparison to most enzymes we have covered that typically mediate one reaction, fatty acid synthase is super interesting because it catalyzes 7 reactions on its own to produce one fatty acid. This explains the numerous subunits it has. So, given its many subunits and associated reactions, I believe that the best way to understand this process is to consider what happens both with the reactions and the protein. To do so, I want to consider this rendition of fatty acid synthase. For simplicity, only one half of the protein is shown, and every different block of color represents a subunit. Obviously, this illustration is much simpler than what I just shown, as the subunits are not presented in the correct order and the linker subunits are not shown. Nonetheless, this should still be helpful to visualize the reactions a little better. Broadly speaking, fatty acid synthase can be divided into two functional domains: a condensing domain represented by the blue subunits, and a modifying domain represented by the hot colors. As we get into the reactions, it will get clearer why they're called that way. Now, to coordinate all the different reactions occurring between the condensing and modifying domains, the fatty acids will be shuttled between two main sites: the cysteine residue on the Ks subunit and the acyl carrier protein, or simply ACP. If we consider the structure of fatty acyl-CoA in these two sites, you will see that they all have in common this thioester bond that links to the fatty acid. In other words, all the fatty acids are connected to their carriers via sulfur. Additionally, you can notice that both ACP and CoA have this phosphopentadiene segment, which is super important as it carries the fatty acid around. As such, until the fatty acid is completed, it will stay bound to either ACP or the Ks subunit throughout the synthesis. So, the general idea behind fatty acid synthesis is somewhat comparable to what we have seen previously with beta oxidation. If you recall, the goal of beta oxidation was to degrade the fatty acids into acetyl-CoAs by removing 2 carbons at every round. Similarly, with the synthesis, the process will occur in successive rounds in which 2 carbons will be added to make the fatty acid chain. So, assuming that we want to make one 16-carbon saturated fatty acid, the starting material for the first round will be one acetyl-CoA and one malonyl-CoA. The process begins in the condensing domain where MAT catalyzes the binding of acetyl-CoA to ACP. As such, this removes the CoA and now we have acetyl-ACP. Then, the subunits MAT and Ks work together to swap out the acetyl group with malonyl-CoA, and that places acetyl on the cysteine of the Ks subunit. The more scientific name for MAT, as you can see on the right, is malonyl-CoA or acetyl-CoA ACP transacylase, depending on the action it performs. Now, at this stage, we have one malonyl on ACP and one acetyl on Ks. The next step in the process is catalyzed by beta-ketoacyl ACP synthase, and it occurs in 2 steps. First, malonyl gets decarboxylated, which in other words means that it loses a CO2 group. Then, the acetyl group on Ks gets condensed onto malonyl ACP to produce acetoacetyl ACP. This reaction frees the Ks site. If we consider the carbon count for a moment, you will notice that we started with one 2-carbon acetyl-CoA and one 3-carbon malonyl-CoA, and from this reaction we now have one 4-carbon molecule bound to ACP. The production of acetoacetyl ACP concludes the reactions that happen at the condensing domain. Now, this compound can be moved to the modifying domain, where the goal will basically be to remove the second ketone group, or in other words, this double bond to oxygen, here shown by the red arrow on the right. The first reaction to do so is mediated by beta-ketoacyl ACP reductase and uses NADPH to reduce the ketone group to an alcohol. Then, in the DH subunit, beta-hydroxyacyl ACP dihydrase removes one water molecule from the compound and creates a double bond between the second and third carbon. In the ER subunit, this double bond is then reconverted to a single bond by enol ACP reductase, which uses NADPH to do so again. After all these reactions, we generated one 4-carbon saturated fatty acid that is still bound to ACP. As you can see from the reactions, one round of this process caused the cell 1 malonyl-CoA, 1 acetyl-CoA, and 2 NADPH. To make the complete fatty acid, our 4-carbon fatty acid will return to the Ks subunit where it will meet a new malonyl that is anchored at ACP. Here again, Ks will mediate a decarboxylation and condensation that will produce a 6-carbon molecule. This 6-carbon molecule will then go through the same steps in the modifying domain to remove the second ketone. This process can then continue for several rounds until the fatty acid reaches our desired 16-carbon count. The final step of this entire process is mediated by the TE subunit, also known as thioesterase, and performs the cleavage that releases the fatty acid from ACP. In total, to make a 16-carbon fatty acid, it requires 7 rounds of synthesis, which will cost 7 malonyl-CoA, 1 acetyl-CoA, and 14 NADPH. Since malonyl-CoA is made from acetyl-CoA and that process costs 1 ATP, we can say that the net cost will be 8 acetyl-CoAs, 7 ATPs, and 14 NADPH. Now that we have this reaction mechanism in mind, I want to get into the regulation of this entire process. As I mentioned previously, the rate-limiting step of this entire pathway is mediated by acetyl-CoA carboxylase. As such, it is a target for many regulatory mechanisms. If we first consider allosteric modifiers, citrate stimulates acetyl-CoA carboxylase. This should make sense since citrate is the vehicle that brings acetyl-CoA into the cytosol. Accordingly, accumulation of citrate forces the production of malonyl-CoA and thus boosts the production of fatty acids to meet the supply of acetyl-CoAs. In terms of inhibition, acetyl-CoA carboxylase gets inhibited by long-chain fatty acids, which should also make sense as they are the final product of this process. The rest of the regulation comes from hormonal control by insulin, epinephrine, and glucagon. Since we'll have a section dedicated to the specifics of these hormones later in our discussion, here I will simply provide an overview of their effects on fat metabolism. If we start with insulin, it is generally seen as a signal that indicates to the body that nutrients are present and the tissues should absorb them. Consequently, it promotes the storage of these nutrients, which, with respect to acetyl-CoA carboxylase, stimulates the enzyme to promote fat synthesis and storage. When it comes to epinephrine and glucagon, the two hormones are quite different from each other and are released in different contexts. Epinephrine is released during exercise or stress to activate muscles, while glucagon is released during fasting or starving conditions as a signal to the liver that the body needs energy. In both cases, the two hormones signal to the body that nutrients need to be used to fuel the body. Accordingly, the two hormones inhibit the enzyme such that fatty acids can be degraded for energy. Another consequence of this inhibition is the decreased production of malonyl-CoA. If we consider back how fatty acids were transported into the mitochondrion, you might recall that malonyl-CoA was basically the sole regulator of fatty acid degradation as its accumulation prevented transport. As such, epinephrine and glucagon not only reduce fatty acid synthesis, they also increase the degradation by removing the brakes that make fatty acids get into the mitochondrion. Finally, as a last quick detail on this process, the cell does not use only 16-carbon saturated fatty acids. It contains additional enzymes such as elongases that are able to increase the chain length and desaturases that are able to make double bonds and turn the saturated fatty acids into unsaturated ones. In this discussion, I will not go over the mechanism of these enzymes, but I wanted to mention that further processing is possible. Now, when the fatty acids are ready, they can get packed in triacylglycerols in a process called lipogenesis, which we are now ready to consider. To understand lipogenesis, I actually want to start with a quick reminder of lipolysis, which is the process by which triacylglycerols are degraded into fatty acids. Having this side of the equation will be relevant later when we discuss regulation. So if we start from triacylglycerols, there are generally two pathways for degradation. The first one occurs in the adipose tissue and liver cells and is mediated by the enzymes adipose triglyceride lipase, hormone-sensitive lipase, and monoglycerol lipase. Each of these enzymes are responsible for removing one fatty acid chain until the glycerol backbone is the only piece left. The other pathway is mediated by lipoprotein lipase and pancreatic lipase, which are expressed in the bloodstream and gastrointestinal tract respectively. These two enzymes directly remove two fatty acid chains from the triacylglycerol and directly produce a monoacylglycerol. The products of this reaction can then be absorbed by either tissues from the blood or the body from the gut. Now, when it comes to the anabolic side of this process, you may recall that generally speaking, anabolic reactions require an input of energy to happen. Consequently, lipogenesis is an energy-demanding process, which makes sense as it usually happens when nutrients and energy stores are plentiful. This energy aspect is directly reflected in the substrates that are required to make triacylglycerols. Instead of directly incorporating the fatty acids back into monoacylglycerol or diacylglycerol, the fatty acids first need to be charged with energy. The reaction to do so is mediated by acyl-CoA synthetase and uses ATP and coenzyme A to make acyl-CoA. Remember that this same reaction was essential for the transport of fatty acids into the mitochondrion and also to generate acetyl-CoAs during beta-oxidation. For convenience in this diagram, fatty acids will be noted as Fa and acyl-CoAs as Ac. Now, just like lipolysis, there are two main pathways to make triglycerides. One that happens mostly in the digestive system, and the other that happens in both the liver and adipose tissues. Let's start with the pathway in the digestive system, as it is the most straightforward one. Simply put, enterocytes, which are cells of the gut, express monoacyl and diacylglycerol acyltransferase, and these two enzymes catalyze the addition of a fatty acid from acyl-CoA and start from monoacylglycerol to remake triacylglycerol. The second pathway, which again happens in the liver and adipose tissue, is a little bit more complicated and starts from the glycerol backbone rather than a pre-existing monoacylglycerol. For the same reasons as why we use acyl-CoA rather than fatty acids, the true starting point for lipogenesis is glycerol 3-phosphate. There are two ways to obtain this glycerol 3-phosphate, either directly by the phosphorylation of glycerol by glycerol kinase or by the reduction of dihydroxyacetone phosphate. This reduction is mediated by glycerol phosphate dehydrogenase and uses NADH to do so. A very important implication of this starting point is that the metabolism of the most important sugars in our diet, so glucose, fructose, mannose, and galactose, directly supply DHAP, glycerol 3-phosphate, and glycerol, as you can see from the red arrows. As such, not only do these sugars provide the acetyl-CoA that can make fatty acids, they also provide the glycerol backbone that serves to store fatty acids. This relationship illustrates how sugar intake is so closely related to fat production. In general, glucose and fructose are the main contributors to this since they are the two main sugars that we consume. Now, when the glycerol 3-phosphate is available, the liver and adipose tissue express GPAT and AGPAT, for which you can see the complete names on the left. These enzymes together take two acyl-CoAs and make phosphatidic acid. As you can see, phosphatidic acid is basically diacylglycerol but with an extra phosphate. Beyond being an intermediate for triacylglycerol synthesis, phosphatidic acid is super important in the cell because it is the starting point for the synthesis of many phospholipids that are then used in various contexts like cell membranes, signaling molecules, and so on. Nevertheless, to go from phosphatidic acid to diacylglycerol, the enzyme lipin, also known as phosphatidic acid phosphatase, is responsible for the removal of the phosphate. Then, DGAT can complete the synthesis by adding the remaining acyl-CoA. Broadly speaking, this completes the picture for how triacylglycerols are synthesized and degraded. The last remaining aspect that I want to add to this is the regulation surrounding this process. As it is conceived now, hormonal control is the main regulator of this process, with insulin, epinephrine, and glucagon being the main actors. The principal target in this regulation is, unsurprisingly, hormone-sensitive lipase. As you might expect, based on what we just covered previously with fatty acid synthesis, epinephrine and glucagon stimulate the enzyme, and this should make sense since HSL promotes the usage of fats for energy. Some of the sources that I have used to make this video also mention that ATGL is a target for epinephrine and glucagon, but HSL is really the main one. Inversely, when it comes to insulin, this hormone inhibits the effects of epinephrine and glucagon, and thus inhibits the enzyme leading to the promotion of fat synthesis. Together with fatty acid regulation, insulin promotes the net synthesis of fatty acids and triglycerols, whereas epinephrine and glucagon promote the net degradation. Alright, back to this overview of fat and carbohydrate metabolism. We have now covered all the pathways that I wanted to go through. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.